Wonderful. I started recording.
Okay. Well, thank you very much, Daniel, for the invitation. My name is Melanie Gillingham, and I'm a professor at Oregon Health and Science University. Uh, Couple disclaimers. I'm not an ophthalmologist. I'm really a nutrition scientist by training and focusing on energy metabolism. And I have been studying this group of rare disorders in fat oxidation, the fatty acid oxidation disorders. And my lab has a lot of interest in LCHADD chorioretinopathy. So that's what I'm going to be talking about today. And assume that many of you are not familiar with these disorders. I'm going to start with just telling you a little bit about what they are. Um, so hopefully my slides are going to go forward. There we go. So long chain 3-hydroxy-acyl-CoA dehydrogenase deficiency is LCHAD, and it's one in a whole family of fatty acid oxidation disorders. There's probably about 20 different disorders in the pathway. Been included in the newborn screening of in the United States since about 2004, and all 50 states screen for this disease in newborns. There's a lot of symptoms of LCHAD that are very similar to other disorders in the family, especially the long chain disorders. They have these episodic metabolic decompensations. So when they're well fed and rested, you wouldn't tell that they have a disease. But if they have to rely on fat, so they are fasting or exercising for long periods of time when they're older, they can go into metabolic crisis. So, for instance, they frequently have cardiac symptoms, hypertrophic dilated cardiomyopathy, heart failure, arrhythmias, sudden death. They can have liver symptoms, hypoketotic hypoglycemia in infancy is a pretty classic sign of LCHAD deficiency. And they definitely have muscle involvement. So, recurrent rhabdomyolysis is a pretty frequent reason for hospitalization in these patients. And that's true for all the long chain disorders. But there's a couple of symptoms that are unique to LCHAD that do not occur in any of the other disorders. And those include a retinopathy, which we're going to be focusing on today, and a peripheral neuropathy with foot drop and a loss of feeling in the hands and feet. So, what is LCHAD? So, LCHAD is part of the mitochondrial trifunctional protein, which many on the call may be very familiar with. So, just to let you know, mitochondrial trifunctional protein is a heterotetramer. So, there are two alpha subunits encoded by the gene HADHA, two beta subunits encoded by HADHB. And they form this holoenzyme embedded in the inner mitochondrial membrane. And they perform the last three steps in fatty acid oxidation. So, one round of fatty acid oxidation will take a long chain fat and make one acetyl-CoA and a chain shortened fatty acid by 2 carbon units. And it starts with VLCAD as the first step to add a double bond between the second and third carbon. Then you have the hydratase as part of TFP alpha that adds a hydroxyl group on the third carbon. LCHAD is this second enzymatic function that creates this double bond O, and then the last thiolase cleaves it to acetyl-CoA, the chain shortened fatty acid, and we repeat the process. There's a common pathogenic variant in HADHA. We call it G1528C. So it's a G to C transition that just changes one amino acid right in the LCHAD binding pocket. And I would say probably 90% of patients in the United States have at least one copy of this particular point mutation or a pathogenic variant. And then a few other variants will actually cause the degradation of the mRNA and a decrease in protein. And we call those patients TFP. So they lose all three of these functions. And it's really the LCHAD patients that have the most eye involvement, the most progressive retinopathy. And if anybody has questions, please feel free to stop me as I'm going along. So we started newborn screening. So has that helped? And I want to at first emphasize newborn screening has been a game changer for these diseases. So we get a blood spot on a filter paper card for all newborns born in the United States. And there was a paper a while ago that looked at symptoms in LCHAD patients pre-newborn screening. So that's the gray bars, the clinical diagnosis versus after newborn screening. So we've decreased cardiomyopathy, we've decreased hypoglycemia, we've decreased hypotonia. It's been a lot better for these babies. But the one thing that has not changed is it does not change or halt the progression of the chorioretinopathy. I'm hearing a noise.
I've just muted them.
Okay. So how do we treat these diseases? So we're trying to suppress fat oxidation. So all the patients are advised to avoid fasting. So eat small, frequent meals throughout the day. We put them on a low-fat diet. The concept there is if you can't oxidize long-chain fat, let's restrict it in your food. So it's recommended that they consume 10% of their calories from long-chain fat, which is extremely low and difficult to do. We supplement them with medium chain fats to bypass the long chain pathway and give them a little bit of extra energy. And I'm showing you some of the infant formulas and some of the supplements that patients can take. We monitor for vitamins, essential fatty acids, and some people supplement with carnitine, although I will say that's really controversial. Again, treatment improves, decreases episodes of rhabdomyolysis, improves the metabolic decompensations, but it has not halted or stopped the progression of the retinopathy. So for the rest of the talk, I'm really going to describe what the chorioretinopathy of LCHADD looks like and what its progression is across the lifespan. And my hypothesis is that the retinal pigment epithelium is the initial cell layer to die, resulting in later loss of photoreceptors and decreased vision. I do think that's still a hypothesis. I'd love to really put a pin in it and prove that definitively. Um, I'm going to try to identify factors associated with the progression among patients. And again, our hypothesis is that the three hydroxy fatty acids that build up as a result of the loss of LCHADD function are toxic, specifically to the RPE. And that is a hypothesis. We've done a lot of correlations, but I don't know that we've definitively proven that. I'm going to explore LCHADD RPE phenotype using an in vitro model of iPSC-derived RPE. And our hypothesis there is that RPE cell death is not really due to the energy deficiency, which has been one of the hypotheses in the field, but related to lipid peroxidation in the presence of three hydroxy fatty acids. And then at last, I'll describe some of the effects of antioxidants or gene addition on the RPE phenotype. So I mentioned that LCHADD is the only fatty acid oxidation disorder that has a retinal chorioretinopathy and a progressive vision loss. The other thing that's unique about them is this specific species that we detect in blood. So patients with LCHADD have high 3-hydroxy acylcarnitines is what we test. So those newborn blood spots are screened for acylcarnitines. And I think of acylcarnitines as opposite of glucose, but they go up and down with feeding, fasting, exercise. So they're changing constantly in blood. But it's really if you have an intact hydratase, so you have a normal hydratase function and the loss of the LCHADD function, you get this fatty acid with a hydroxyl group on the third carbon that then will come back out into blood. And that's how we diagnose patients. This is a study we did where we have 6 patients who have 2 copies or homozygous for G1528C. We have 6 patients, maybe it's 4, I don't remember exactly the numbers, of heterozygous, so compound heterozygous, and 3 patients with TFP in the green. And so we fasted them overnight and drew blood, and you can see that after you fast and you mobilize lipids from your adipose tissue, then you have higher hydroxyacylcarnitines. We fed them a standardized breakfast, and 2 hours later, they dropped by about 60%. And then in the afternoon, we did a 45-minute treadmill exercise, and there really wasn't much of a budge. So it's really the feeding and the fasting that seems to dictate how much of these are in your blood. But I'll point out that if you lose all 3 functions, your hydroxyacylcarnitines are quite a bit lower in the TFP patients. So if you knock out the hydratase, you don't get these species as high in your blood.
Quick question. How are they eliminating them from blood?
How are they eliminating them from blood? Um, that's a good question. I think they're clearing. There are some that get peed out as carnitine esters in urine. So I think of this 60% drop in acylcarnitines as a decrease in production. So the production of the hydroxyacylcarnitines parallels the free fatty acids. So as you're mobilizing adipose from your lipids, and you're trying to burn them, that's when they go up. And so it's really a drop in production that we're seeing when you eat and you suppress lipolysis because insulin's released and you're not mobilizing fat. Does that make sense?
Absolutely. Yeah. I just, without the thing that gets rid of it, how does it go away was basically but,
Right.
yeah, urine is reasonable. Is
I don't
there
know that
sort
it
of,
all is urine. I think maybe there's some residual enzyme activity. We don't think nobody has zero enzyme activity. So there's probably, a little bit. When I've done stable isotope studies, they have about 10% normal activity. So they are clearing it slowly over time. But I think this drop is really a decrease in production.
yeah, thank you.
Yeah, it's a good question. I don't know that we know for 100% exactly how they're cleared. So that's something we should think about. Okay, so we recently wrote a paper on the stages of chorioretinopathy. This was originally published a number of years ago by Tina Tiny, and we sort of updated it with some newer technologies. So I'm going to walk us through this kind of slowly. I will confess I'm not an ophthalmologist, so if anybody has any other questions, please go ahead and pipe in. So stage one is pretty normal fundal imaging, and this is fundal autofluorescence. So we should see nice smooth white, which is the RPE reflecting the light back, and that's what you see here, and very well-defined layers of the retina in OCT images with normal ERG response, both dark and light adapted. Stage two, we start seeing that modeling and it usually starts centrally. And we start seeing some loss of the autofluorescence, suggesting loss of RPE kind of in the middle, and maybe a little, still pretty normal OCT, but we're starting in normal ERGs. They haven't really declined yet. 2B is a new stage that we define. We're starting to see a little bit more. We're starting to see some disorganization in the OCT images, although the ERG is still preserved. By stage three, you're really seeing a lot of changes in both autofluorescence and fundal images. And now we've sort of lost the definitions across OCT, and we start seeing some decline in ERG response. By 3B, we're starting to see loss of the RPE layer in these yellow arrows in the OCT image, very flat ERGs. And by 4, there's really not much defined retinal layers and no ERG response. So that's sort of how it progresses as the patients age. So which cell
Sorry.
dies? Yeah.
Oh, I just wanted to ask about the sort of the age correlates here. What is there sort of a general range of ages that
Yes. I will show
work?
you yeah, I'm definitely going to go over that. So
Okay.
Um, they have relatively normal preserved vision up till about puberty, but by most of my patients that are in their mid 20s to late 30s can't drive, have no night vision, so they lose their vision starting at puberty, and it seems to be steadily declining. Now that being said, I don't have any patients that are 40 or 50. This disease was described in 1990, so the oldest patient I have in my current cohort is 36. I don't know what a 50-year-old with LCHADD looks like. So we will see as they age what the phenotype becomes as they get older.
Thank you.
Yeah. So this is a slide courtesy of the genetic ophthalmologist that I work most closely with, Mark Panisi. We've had these debates back and forth. What's the cell layer that dies first? And he argues based on, if you look at fundal images of LCHADD, that it looks similar to choroideremia, gyrate atrophy, which is a loss of the RPE choroid before the photoreceptors. And it doesn't look like retinitis pigmentosa where you lose the photoreceptors. So he's arguing that this suggests that it is, in fact, the RPE that's the first layer that dies in LCHADD deficiency. So we have a natural history study that we're just tying up the last bits of data for this study, where we enrolled 40 patients, and they were seen at either my institution at OHSU or the University of Pittsburgh. So patients would fly to our institutions. They'd have a series of 2 or 3 days of testing, including all the photos that I just showed you. We'd get a fasting blood sample. So we'd have them come in first thing in the morning after an overnight fast and get a blood sample to measure acylcarnitines. And they did a diet record. They flew home, and then we repeated it 2 years later. So we're really just trying to characterize the change in vision and retinopathy over the lifespan. So we enrolled 40 patients in this study. My best guess estimate is there's probably between 400 and 500 patients with LCHADD or TFP in the country. So it's about 10% of the population. Um, we stratified them by age. We really wanted to capture vision across all ages. So we had 2 to 7 year olds, 10 in the 8 to 14 year olds, 13 between 15 and 21, and 9 that were greater than 21 years of age. Here I'm showing you whether they were picked up by newborn screening, or they had a family history, or they showed up symptomatic as a newborn in the emergency room. We have a nice mix of heterozygous versus homozygous with the common mutation, the G1528C. However, there's very few TFPs. There's not that many of them in the country, so we only have 3 that had no copy of G1528C. So we wanted to ask what factors are associated with chorioretinopathy. So my statistician, Dr. Cho, we were looking at, does early diagnosis slow down the progression? What's its impact on visual function? And what is the impact of genotype on retinal function? So our outcome variables were visual acuity, visual fields, and our predictor variables were sex, age, newborn screening, or presenting symptomatically. So assuming that they had more episodes of decompensation, and genotype. So this top row is always going to be the picked up by newborn screening or family history row. And the bottom row is the patients that presented symptomatically. So were very sick as infants and had multiple decompensations before we made the diagnosis. And I tried to match them for both genotype and age. So you can see the difference. The bottom row clearly has much more loss of pigment. This is 18-year-old up to 30, and you can really see the progression with age, but also how much more severe the symptomatic patients were compared to those who were diagnosed by newborn screening or picked up because of family history predominantly, because these are a little bit before newborn, or a little older than when newborn screening started. The same thing is true with the fundal autofluorescence. So if we're assuming this is representative of a loss of RPE, you can see that those who were symptomatic have a loss of RPE starting at 18 that's pretty encompassing all of the central macula, and then it's getting worse as they get older. Same thing with OCT. You can see pretty normal are layers of the retina, and as they get older, they start becoming more and more disorganized. It starts earlier in the symptomatic patients, and so you just kind of lose that RPE in the normal structure of the retina. And by 30, most of them have pretty severe vision loss. So if this is looking at their visual acuity by logMAR, I put the Snellen equivalent sort of over here to the side. The orange are the symptomatic patients, and the blue are the picked up by newborn screening or family history. And I've just noted the TFPs in green. And in our cohorts, TFP patients who have those loss of all 3 of the functions of mitochondrial trifunctional protein and lower hydroxyacylcarnitines appear to maintain normal vision. Now, I say appear, I mean, there are some changes in ERG. They do have slight pigment changes, so it's not 0, but they, at least out to about 30, they seem to have normal vision. But it's definitely lower in the symptomatic patients, and that's true for both rod responses in the ERG as well as cone responses in the ERG. The symptomatic patients are always lower. So the early diagnosis and the institution of our current therapy seems to slow it down a little bit. It also is really apparent that it's progressing with age, regardless of if they were picked up by newborn screening or if they presented symptomatically. So as they get older, their vision's going down. It just seems to be a little bit slower if we diagnose them early. So another question we've been curious about is, could we find any correlation between those 3 hydroxyacylcarnitines, which are unique to LCHADD, and the retinal visual acuity or the some of our visual outcomes. So we collected these overnight fasted blood samples in 39 of the 40 participants. So we had one where we just couldn't get venous access. We analyzed them for acylcarnitines, and we did untargeted metabolomics and lipidomics just to try to see if there's other biomarkers that might correlate with vision. And a postdoc in the lab, Leah Chung and Dr. Cho, my statistician, were really spearheading this look at what's going on from a blood perspective, and can we correlate that with vision? So we were asking the questions, are acylcarnitines related to genotype or visual acuity, and are there any other metabolomic markers unique to LCHADD? So when you look at the principal components from acylcarnitine, so this explains most of the variation in the acylcarnitines among those 39 patients, it does have an association with genotype. So the mean principal component for the patients who are homozygous for the common mutation is higher than the trifunctional protein. You can see there's a huge variation in the heterozygous group. And I suspect that that has to do with what the other allele is doing to the protein. And we just haven't been able to try to narrow down what's causing this a big variation. But I suspect patients down here have a deletion or something that's more severe, and patients up here have maybe a milder mutation on that other allele. Interestingly, this just one blood sample correlated pretty tightly with visual acuity. So those who had higher hydroxyacylcarnitines in this blood sample had lower visual acuity. So lower logMAR, higher Snellen. For the metabolomics, we found 56 species that were higher in LCHADD and 46 that were lower. And most of the ones that were higher were these 3-hydroxy fatty acids or the 3-hydroxy acylcarnitine. So it's basically mirroring the acylcarnitine profile. So we see, 3-hydroxy stearate, 3-hydroxy oleate. It looks very similar. Probably more surprising was the decrease in complex lipids. So there's a decline in phosphoethanolamines, sphingolipids, sphingosines, those types of things. And our principal component analysis really can separate out the control population compared to the LCHADD population. So again, it looks like the metabolomics mirrors the acylcarnitine profiles. When you look at, the acylcarnitines versus the metabolomics, they correlate really tightly. And the metabolomics, those 3-hydroxy fatty acids, if you do the PCA again, it matches genotype. It correlates with vision. Previously, we've seen that in one publication, I reported that it correlated with long chain fat intake, but that was not the case here. It was really just sort of unique to the genotype and correlated with vision, but was not related to their dietary intake of fat. We did look to see, are they following the recommendations? Is this having any impact? And so this is grouped by this different age buckets that we recruited patients in. And as you can imagine, the little ones whose parents are in charge of their diet are following it pretty good. They're eating a really low fat diet. And as patients age, their fat intake is creeping up. But the average was 14%. And again, the average dietary intake for fat among Americans is about 38. So they're eating a pretty low fat diet. The calories from MCT or trieptanoin C7 is kind of variable. So some patients are, consuming quite a bit, and some patients are consuming very little. It's kind of all over the board, but there wasn't a difference across the age groups. So I think they're doing what we're asking them to do, avoiding fasting, eating a low-fat diet, but all of our current interventions are not stopping the progression of vision loss over time. We looked at lipids, and I was kind of expecting to see some bigger differences in lipids, but we didn't see huge difference in circulating lipids, which was somewhat surprising. The total amount of lipids in their blood was lower, and I think that's a reflection of their low-fat diet. We saw some increases in some simple triglycerides, but only 34 species. Again, decreases in complex lipids like phosphoethanolamines, but their proportion of fats, polyunsaturated, monounsaturated, saturated, are pretty consistent. They're the same as the controls. So I really think what happens is that they're following this low-fat diet and their total lipids are lower, but the proportion of those fats are kind of maintained. So here's the data. This is brand new. Haven't really shown this other than our group, and we're tying down the 2-year data right now. So this is change in visual acuity in our 40 patients across our natural history, and I haven't sent it to the statistician yet. But if we just sit and look at, again, we've got age across the top. You can clearly see in the yellow boxes the homozygotes are having increase, decrease, a bigger change in visual acuity, and it's occurring in these older ages. The trifunctional protein, again in green, it actually goes up a little bit. Mark tells me this is just the normal variation of repeated visual acuity testing, and he would just call that good vision on both visits. But you can see it's kind of flat. So they seem to maintain their vision between 2 and I don't know, we're starting to see some changes here in late teens, early 20s. And it's not really into this second decade of life that we're really seeing this decrease in vision. There are some notable exceptions in the data set that are losing vision a little bit earlier, but that's sort of how the progression seems to go. They seem to maintain it pretty good, and then towards their late teens, 20s, we start seeing that decline in vision.
Hey,
And I do,
Melanie,
yeah,
sorry to interrupt, but have you
that's
tried
okay.
stratifying your population by sex? I know you don't have a lot of patients, but I'm wondering if there's any sex-related effects here.
That's a great question. In our previous study, we always have included sex in our models, and sex has never been a significant factor. I think it becomes a significant factor occasionally on one or two of the ERG, like the speed of the rods or cones, but it doesn't seem to be a consistent factor. Um, we do see sex effects in the cardiac complications, but I haven't seen strong sex effects in the eyes, but that's a good question. So here's another piece of data that I just got. Again, we've, as a group, have been debating this. I was really hoping we could take some of the OCTA. So we have a really advanced OCT angiography lab where they can take these slabs and separate out the different layers of the retina. And so I asked them to calculate what layers decreasing the fastest, right? Is it the choriocapillaris, the RPE, or the EZ zone, or the photoreceptors? So this is one patient. They've done about 3 or 4, and we see the same pattern. I'll have you focus just on the table over here. So the change. So we have the baseline across the top, and then 2 years, and then she's sort of overlaying the changes that happen over those 2 years. And this indicates that the choriocapillaris is declining the fastest, about 50% loss over 2 years, the RPE about 40%, but less in the EZ or the photoreceptor layer. Now, I will say Mark cautioned me in interpreting this data. Again, I'd really like to prove that it's the RPE that's the first layer, and this data would suggest maybe it's the choriocapillaris. But the choriocapillaris is flow, and these other are more OCT images. So exactly if it really is the choriocapillaris versus the RP, I think is still an open question. But at least based on OCTA, it appears that the vessels are receding, and then the RPE, and then the photoreceptors. So we're still trying to work out through all this data which layer of the eye is declining the fastest.
Can I ask a question?
Sure.
On your SLO, have you tried to do a near infrared? Because that's supposed to be more directed to melanin. Maybe you would get, different.
Near infrared. I don't recall, but I will go back and ask my ophthalmology team if we've done that. That would be a great idea.
Yeah.
Write that down. I feel like we've done every test possible, but I don't remember them telling me that we've done near infrared. So I will ask.
Okay.
Thank you. Okay.
And while we're paused, sorry, Tom Levin also has a question in the chat.
Okay. I don't see the chat. Can you?
Oh, okay.
Melanie, I was just asking, to prove your hypothesis, a couple of us on here know Mark very well. Have you looked at your ERG C wave early on in patients with early stage disease to see if your C wave changes prior to any of your other, I saw you showed scotopic B wave, and I think you showed cone flicker, but you looked at the C wave. And likewise on these OCTAs, you're not measuring flow, you're just measuring flow voids on your choriocapillaris, correct? like these
That
large
is,
low void?
yes, I believe that is correct. I'm thinking about the data that I get from the full field ERGs, and I'm wondering if I see a C wave. I know we've been very diligent about doing that in the mice, but I'm trying to remember if I capture that in the ERG, but I will go back and look. I'm thinking I don't know that I see it on the report, but I will go back and look. But that's a good point. C waves should be able to tell if the RPE is going faster.
Yeah, it's not always the easiest to pull out, obviously, especially if these patients are younger, but it's something to consider.
Yeah, and in some of the little ones, we just don't have
Yeah,
good
no chance.
ERGs. Yeah, the 2-year-olds just aren't gonna cooperate. But their vision is pretty normal, so it's really the older ones that we want to capture anyway. So, good points. I'll look for C wave and the infrared. So thank you for those suggestions.
I mean,
I'm
are
hope
these your near infrared images though on your left side from your OCTA?
Yes, you think these are near infrared?
Typically, you get a near infrared.
But you're
Okay.
probably
OCTA.
referring to near infrared autofluorescence, which is used for melanin.
Yeah.
But this is near infrared reflectance, which does not pick up on melanin. This is the standard scout image for OCTs. I was
Okay.
just going to say, I mean, of course, Mel, I've chatted with you before and I've looked at the work. I feel like I don't need any convincing that this is an RPE primary defect. That clinically, everything that you're showing is extremely consistent with, a primary RPE problem. You may want to take a poll of, a bunch of retina specialists and say whether any of them feel, that there's significant doubt. I don't know if other clinicians on the line would argue differently. I mean, but for me, this just looks
You're
very
convinced.
obviously. This obviously looks like an RPE defect with a tight choroidal degeneration, very akin to choroideremia.
Okay. Good. Well, that's good to hear. I mean,
I mean, that same argument, Jason, was made for choroideremia for a long time until they kind of figured that out too.
So
I
what
think
do you mean? What do you mean, Tom?
The same argument was made for choroideremia, right? Trying to figure out if it's choroid versus RPE for a long time.
Yeah.
But I don't disagree with you, Jason.
Yeah.
I will say, I've always thought it has to be RPE because when they've done, where is TFP expressed? Well, it's expressed in RPE and photoreceptors, but not necessarily in the endothelial or choroid layer. So it seems odd to me that a defect of a protein that's not even that cell would cause that layer to die first, which seems to be another argument for why the RPE is more likely. But it's good to hear.
There's another much rarer gene defect, PNPLA6, which phenocopies choroideremia, and it's the same argument. I mean, because it's so rare, people are now trying to figure out, is it the RPE or the choroid, because you get both degenerating. But it's the same thing. The expression level is just markedly higher in the RPE than it is in the choroid tissue. Anyhow, early fundus autofluorescence changes are usually a pretty sensitive marker that the problem is
RPE. Okay, good. Well, thank you. That makes me feel more confident that we're on that right track. Okay, so sort of summing up the human data, the photoreceptor looks like it's a secondary loss of photoreceptor degeneration is secondary to the loss of RPE. The retinal changes are more severe in LCHADD than TFP. At least in my hands, the TFP patients are maintaining their visual acuity pretty well. Again, the oldest I have is 36, so I don't know what's going to happen when they're 50. And early diagnosis and treatment has slowed it down. It really does make a difference. However, it's still progressing with age. And elevated hydroxyacylcarnitines definitely correlate with visual acuity. But that's correlation. It doesn't prove cause and effect. Mark really challenged me. We needed a preclinical model to kind of test this. So that's when we went to modeling LCHADD retinopathy in cell culture. So we had lots of skin biopsies from diagnosing patients pre-newborn screening. That was the standard of diagnosis where you get a full thickness skin biopsy and measure fatty acid oxidation in fibroblasts. So we took those fibroblasts, dedifferentiated them to induce pluripotent stem cells. And then I had a talented postdoc at the time who pushed those to RPE. So in this top panel, you can see that the LCHADD iPSC-induced RPE have similar as O1. ATPS best, RPE 65, and here's the bright field. They formed this nice cobblestone layer like we expected. We looked at the epithelial resistance on RPE grown on trans wells, and it was the same as the wild type cells. And we did TEM to see tight junctions, and again, we saw tight junctions. We expected that the LCHAD cells would express TFP, that it's really just the loss of the LCHAD activity, but the protein would be present. And we did see that. So they have the same level of TFP alpha and beta subunits, as well as VLCAD. So our Westerns show that they do express the protein. And then we checked to see if they could phagocytose FITC labeled bovine outer segments like RPE should. So we added them, and so the green color are the POS that have been internalized into the RPE. The yellow are counterstained with red rhodopsin, so they turn yellow, and that's still sitting on the top of the cells. But you can see there's really kind of similar uptake of the FITC labeled outer segments in both LCHAD and wild type RPE. So it looks like we can form the RPE. They seem to function somewhat like you would expect an RPE to in culture. So we really wanted to know, did they have a defect in burning fat? So to do that, we matured these on seahorse plates, and we looked at the fat oxidation in a seahorse assay. So we have wild type with BSA, or wild type with BSA plus palmitate, which is a C16, a long chain fatty acid bound to BSA. And you can see that in the wild type, you get this nice bump up in oxygen consumption, suggesting that they are oxidizing it, both in the basal and in the maximal respiration, but you don't see that change in the LCHAD group. Now, these conditions were under low glucose with carnitine, so you really have to take the glucose away and add carnitine to push them to burn fat. So that's the conditions that we did these experiments in, and the bar graphs are just showing the means of these grayed areas above. So we get this increase in both basal and maximal respiration when we add fat to the wild type, but we don't see the same rise in the LCHAD cells. So it looks like, yes, they don't burn long chain fat very well. So that's good. It looks like they have LCHAD. This is unpublished data, but I was really curious. I've always wondered why LCHAD and why doesn't VLCAD have a retinal phenotype or CPT2? So we had VLCAD cells that we took through the same process, iPSC to RPE, and looked to see if they burned long chain fat, and they don't. So this is the LCHAD cell line again, and here's the VLCAD cell line. This is just basal. We didn't do the full mitochondrial stress test, but we didn't see an increase in C16 when you in the VLCAD cells like we do in the wild type. So both LCHAD and VLCAD cells don't burn fat, but just to remind you, VLCAD does not have a retinal phenotype, only LCHAD does. So we also look to see if they can't burn fat, do they accumulate it? So this triglyceride assay is just in cultured cells. We haven't added any extra fat, but you see an increase in triglyceride in the LCHAD RPE. And if you do add it and stain for a lipid, you see this C16 has this increased lipid droplets that accumulate in the cells. And then we wanted to look at ketones. So again, we grew them on trans wells, added C16, and you see this pretty dramatic rise in ketone production in the apical well in the wild type. And it increases in LCHAD. It just doesn't quite make stats. Again, there's some residual activity in these cells, so they can do a little bit of fat oxidation, but clearly can't make the same amount of ketones in the presence of fat that the wild type cells do. So again, it looks like they accumulate fat, they don't burn it, and they don't make ketones and secrete them towards the apical membrane. Here's just an example. I found this data. We haven't published this either, but the VLCAD cells do the same thing. They accumulate lipids. And I re-put up the pathway just to remind you. So patients who have a decrease in VLCAD, which is the enzyme just proximal to the mitochondrial TFP enzymes, do not have a retinal phenotype, but they do have lower fat oxidation in RPE in culture, and they do accumulate lipid. So to me, that argues that those two factors are probably not what's driving the retinal phenotype that we see in LCHAD. So do the RPE accumulate the 3-hydroxyacylcarnitines? So again, we fed them either BSA or BSA with palmitate, and you see a little bit of bump in the C16 when you in the wild type cells, but you see these enormous rises in 16-1 and 16-hydroxy, either with or without added C16. So they're clearly accumulating acylcarnitines like you would see in blood and pretty high levels in cell culture. And my naive concept was that they're going to accumulate all these hydroxyacylcarnitines and then they're going to die. And that's what's causing the cell death. And in the presence of glucose, and we add fat and they accumulate these hydroxys, they're swimming in them and they're perfectly fine. They didn't die. So that kind of took us back to thinking about, okay, what's going on here? And led us to sort of the hypothesis, well, maybe we need to stress them a little bit more. We need to have the accumulation of 3-hydroxys, and under some sort of additional stress, we might see some cell death. So we started with oxidative damage. If they have any sort of increase in oxidative damage, would that stress then lead to cell death? Initially, we tried hydrogen peroxide. And in glucose conditions, all the cells did fine. But when we lowered the glucose, added carnitine and fat, the wild type cells actually survive better with hydrogen peroxide added, whereas the LCHADD cells quickly died. So it seemed like that added oxidative stress in the presence of fat as their energy substrate really decreased the viability of these cells. As we were talking, yeah.
To that point, like based on the phenotype, right?
Mm-hmm.
Like over time, you're also getting delayed choroidal perfusion, you're getting loss of choroidal perfusion, right? So their glucose concentration may not be the same as it was prior to your stress point. Just something to think about, right? It's probably not the same over time. And so they may not be as happy as yourselves in culture.
You're right. Yeah.
Yeah. I have a half question, half point.
Yeah.
So fatty acid oxidation classically makes NADPH. NADPH classically fuels the antioxidant defense, at least the glutathione-dependent antioxidant defense. And that would also not be available in the VL, the other disease.
Right.
And so have you tried this same experiment in the other disease?
No, we need to do that. We need to do the full set of experiments with VLCAD, LCHADD, and TFP cells, and we're working on that. But you're right. That would be a great proof of concept if we did the VLCAD cells and did the same experiment to see if they have the same oxidative stress response.
My expectation would be that it is kind of also, I guess also another question, how much H2O2 did you use? And does that correlate well with anything that we might encounter in a living organism?
No, it was very artificial. I don't know the exact amount off the top of my head. I think Tiffany treated each of the cell lines to see what what kind of dose that they tolerated.
Yeah.
But these were high levels of oxidative stress, which again,
which is fine. I mean, it might correspond to like low levels accumulating over time or whatever in a person, but
right.
Yeah, I'm kind of an oxidative stress skeptic, as some people know and many people disagree with. But and so that's why I'm interested in seeing what the other cells would give you.
Melanie, I think I had asked you this question before and I just forgot the answer. What about exogenous addition of hydroxyacylcarnitines into wild type cells in comparison to the same amount of a regular acylcarnitine, sort of addition and doing a tox set of tests without any other stressor?
So that was my original idea, was that I was going to add hydroxyacylcarnitines or hydroxy fatty acids and the cells were going to die. And in the wild type, they have an intact LCHADD and they just burn them and they disappear within less than 24 hours.
Got it.
They go in.
They're still able to get into the mitochondria through the carnitine transport system.
I'm assuming they're imported, made into a long chain acyl-CoA, transported through the CPT system, and are oxidized. So by 24 hours later, the hydroxys are gone in wild type cells. They don't accumulate. And I'll show it a figure we haven't published yet, but that where we did We incubated and then took that media off the LCHAD cells and put it on wild type, and they cleared them. So wild type, they don't
just
accumulate. Yeah.
So, and I guess we're getting even more artificial, but just mostly out of curiosity, if you put the hydroxyacylcarnitines on with atomoxir so that they don't have access, what happens then?
That's a great experiment. We have not done that. If we took the wild type and add atomoxir and then add the hydroxys, that would be a really lovely
Yeah.
proof of concept again. That would be a
Like
nice
putting
experiment.
you can buy the acylcarnitines and the hydroxyacylcarnitines both with atomoxir, so you're not burning through any of it and getting a sense of relative toxicity of the two. Of course, if the tox is in the mitochondria, you're missing it. But if the
Right.
tox was, elsewhere, I mean, mechanistically, I think, like I said, I forgot if we talked about this, but this sounds so similar to Smith-Lemli-Opitz, where you have like the precursor cholesterol is one of the most highly oxidizable sort of, sterols available, and the mechanism is just this profound oxidation that, you can be an oxidative stress skeptic, but with, some of these that are so highly oxidizable, I think like when in Smith-Lemli-Opitz, I think it's been pretty well proven that that's, the major
mechanism.
And so I'm sort of just curious if the same thing, an analogous thing is happening here with the hydroxyacylcarnitines.
Yeah, those are all great ideas. We'll have to try that with wild type. Put in atomoxir and add the hydroxys and see what we got.
Well, I want to, I'm sorry, I hate interrupting, and yet I do it all the
It's okay.
time. Maybe I don't actually hate it deep down. So two additional experiments that you can consider. First, growing the cells with a light in the culture.
We've talked about that, doing a light exposure. Yeah.
And then second, not just atomoxir with the wild type cells. I thought Jason was actually suggesting this, that perhaps atomoxir would protect the LCHAD cells.
That would be good. That also would be a good experiment. So do both.
I like that one.
Yeah, that's a good one.
So,
Yeah.
Melanie, I got one more question. So, one thing that we found a long time ago that hydrogen peroxide does is it activates poly-ADP ribose polymerase, and it chews up all the NAD in the cell. And so we can rescue the cells by adding NAD precursors. And I wonder if you're getting some differential effect here, because NAD is a substrate for that dehydrogenase, right?
It is indeed.
So I wonder if you might have you looked at NAD levels in these? Or?
We've not measured NAD directly, but I'm about to show you some data. If we add NAC at least, and sort of by a different mechanism, try to increase the NAD a little bit, it does rescue them.
Mm-hmm.
So, but that would be a good thing to do too, is to measure the NAD in the cells. I'm assuming it's lower in the LCHAD cells, but I don't know the answer to that.
Yeah, because I think cells really start dying when you deplete the NAD levels. I think that's really bad for cells. That'd be worth testing anyway. But it sounds like you're going to talk some about that. So,
yeah. Okay, here we go. So to try to get at a little bit more physiological stress, we tried polyunsaturated fatty acids. So again, in theory, they are more susceptible to oxidation. And in the wild type cells, you can see that if you add DHA, there's no difference in cell viability. But by about 5 days, the LCHAD cells, two different cell lines, died pretty quickly. And when we added arachidonic acid, it took a little bit longer, but we also saw some cell death after adding arachidonic acid. So in the presence of polyunsaturated fatty acids, we see this decrease in cell viability. Again, this is unpublished data. We didn't have the full data set, so we didn't include it in the manuscript, but I find this particular finding super intriguing. When you do the same thing in TFP cells, so these are cells that we got from patients who have no functional protein, you don't see any decrease in viability. So just to remind you, they don't increase, at least in people. We haven't measured the hydroxys in these particular cells, but in people, they don't have the high hydroxys, and they don't seem to be as susceptible to polyunsaturated fatty acids and cell viability.
Was this also in glucose-free conditions or reduced glucose conditions?
Yes. When we do total no glucose, the cells die, but I think it's pretty low. It's like two millimolar, as I recall, with carnitine. You always have to add the carnitine if you're going to get it to oxidize.
So is the mechanism here that at least partially DHA is oxidized in mitochondria?
So I don't know the exact mechanism, but I can hypothesize.
Yeah, please hypothesize, I guess is what I'm asking.
Yeah. So we think that, DHA is not necessarily imported and the primary substrate in the mitochondria for fat oxidation. I suspect it's the straight chains, so the 18s and the 16s that are more likely to be imported, partially oxidized, and then the C16 is going to come back into the cytosol, and that C16 hydroxy could get incorporated in with a triglyceride next to a DHA. And that hydroxy group is then going to react with a double bond in the DHA and cause an oxidation because its presence is close to a complex lipid that's forming in a lipid bilayer or something like that. I don't think it's necessarily the DHA that's getting oxidized directly, but that it's interacting with these hydroxyl groups on the accumulating 3-hydroxy fatty acids in the cell.
We just maybe partially support that. We certainly have lipidomics data in cell culture when you're adding fats at any reasonable amount, 50 to 400 micromolar tends to be a range that most people use, that the vast majority of those fats are not going into, I mean, this is not surprising, but are not going into beta oxidation. They're being, assembled into phospholipids, tags, the whole thing. So there'd be plenty of opportunity for whatever, hydroxyacylcarnosine was created to interact with a DHA and a phospholipid tail or a tag or whatever else.
Right. That's what I suspect is happening. Because I don't think there's lots of evidence in other cell types that DHA is imported into the mitochondria for oxidation. It's used for other complex lipid formation.
I had to look this up real quick because I couldn't remember, but DHA is a major component of outer segments.
Yes.
And in the TSC1 model that we study and that Punzo showed, DHA rescues some of that phenotype when they supplement the animals with DHA. So I'm wondering did you try feeding your cells with like outer segments in a reduced glucose condition to see if that induces cell death in them?
We haven't done that experiment. That would be another really nice proof of concept experiment that it's coming from the photoreceptors. So no, we haven't tried low glucose, feed them the outer segments and see and check the survival. But that would be a good idea to do. I think we still have the outer segments. We could try that. Yeah, good suggestion. I'm trying to write down all these ideas. We got a lot of experiments to do.
Well, you can always rewatch the video if you'd like.
Okay, good. All right. So we wanted to know if this was what kind of oxidative stress was going on. So we did a mito-Ross stain, and you can see that the LCHADD cells with the DHA have increased mito-Ross staining and decreased total glutathione. So again, going back to Daniel's favorite oxidative stress type of response. We also did a BODIPI-C11, which measures lipid peroxidation. So again, the cells that have this more yellow color are the LCHADD cells, and they have increased lipid peroxidation by this assay compared to the more red in the wild type cells.
Melanie, have you looked at ferroptosis markers in these cells?
We've looked at, we tried adding ferrostatin to try to block ferroptosis, and it partially rescues them, but not fully. But we haven't looked at ferroptosis markers directly, just trying to block ferroptosis and see if it rescues. And it does have an effect. It just didn't.
Yeah, I mean, just everything's kind of leaning that way despite Dan not liking oxidative stress, right? With glutathione down, your mito-Ross up. I mean, you can look at labile iron, stuff like that, kind of pointing that way with when you add your PUFA, right? And then you go from
right. Yes, and we should do that. We should measure. We just went straight to the ferrostatin, but you're right. We should measure the ferroptosis type of intermediates as well. So the last thing we tried was we added NAC to see if the N-acetylcysteine would rescue them, and it did increase their viability. We tried a deuterated DHA, which can't be oxidized, and with deuterated DHA, it also was similar to the wild type. So either preventing the peroxidation by adding a DHA that can't be oxidized, or by adding NAC, we were able to rescue the cells to a similar viability as the wild types. So that, again, kind of all points to your point that it's ferroptosis, necroptosis, some sort of oxidative stress that seems to be associated with their decreased viability in the presence of polyunsaturated fatty acids. So the next thing we wanted to try was gene addition, but biochemically that was a little bit tricky because we weren't sure if we added a wild type copy of the alpha subunit, would it find the endogenous beta, go to the mitochondria and form a holoenzyme? So the first thing we did was we did a immunoprecipitation. So this is the vector. We cloned it in a AAV5 with a CAG promoter and a flag tag. So we tagged the flag in this image with VLCAD to see does it go to the mitochondria, and this yellow seems to suggest it does. It's co-localizing with VLCAD. And then we did immunoprecipitation. So we pulled down with flag and then stained for the HADHB, the beta subunit, and we saw it there. So it looks like it is in fact finding the endogenous beta and going to the mitochondria to form a holoenzyme. So then we said, does it rescue the cells? So these are the wild type, the untreated LCHADD in the green are the LCHADD with the construct, the AVHADHA, and it increased the basal respiration. It increased the production of ketones on transwells, and it completely rescued their viability with DHA. So all three of our phenotypes seem to improve with the gene addition approach. And probably most importantly, it completely normalized the hydroxyacylcarnitines. So the hydroxyacylcarnitines in untreated cells are very high in the presence of C16, but almost zero in the vector-treated cells. So it looks like gene addition improved. So I'm going to sort of finish up here. The LCHADD RPE do not oxidize fat. They accumulate these hydroxyacylcarnitines. They have decreased viability. It appears to be a lipid peroxidation mechanism when polyunsaturated fatty acids are present, but adding a wild type copy did seem to rescue them and normalize their acylcarnitines. We're still trying to figure out, as we've talked a lot about, whether it's ferroptosis, necroptosis, what exactly this pathway is. But I suspect it's something like that. So future directions, I'd like to know if it's cell autonomous. We talked a little bit about this. We incubated the cells in low glucose with C16, and then we took some of that conditioned media here, and put it on wild type cells, and they just completely chewed up all the accumulated hydroxyacylcarnitines. So I'm curious if whatever approach we have, we're probably not going to be able to, if we did gene addition, hit the whole retina. If you have a corrected cell next to a wild type cell, if it clears the hydroxyacylcarnitines, does it rescue its neighbor? I don't know. Is it cell autonomous? So that's, I think, an important question for future treatment. We have made a mouse. Mark was really pushing me that we absolutely needed a mouse. So we used CRISPR to put the G1528C point mutation into the mouse, and we're working on a gene addition experiment right now, and maybe some webinar in the future. Myself or someone in my lab can present our mouse data. I'm not going to really go over it today, but it does look like the mouse has the retinal phenotype, so that we at least have an animal model we can move forward with. So with that, I'll just say I didn't do all this by myself. I have a fabulous team of genetic ophthalmologists who see my patients. I have a great lab group. My study coordinator, Ashley, did a great job of recruiting 40 patients. We weren't sure we were going to be able to get that number, but we did. So I have a good support from wonderful people and a lot of funding to thank for all of this data. And I will stop sharing, and I'm happy to answer any additional questions.
Melanie, this is Jason again. This is like, far in the future. You've got so much, so many cool experiments to do before then. But we were just, a group of us had met recently who are also interested in the peroxisome, and were reminded of a paper that came out in which the peroxisome beta oxidation enzyme MFP2 was knocked out and had a really profound RPE phenotype. Okay. And the paper, this was a group in Belgium at KU Leuven. The paper showed that the phenotype of the RPE degeneration could be rescued when you eliminated photoreceptors by crossing it to a a rapid degeneration, photoreceptor degeneration model,
Huh.
meaning that if you didn't burden the RPE with the incoming load of photoreceptor outer segments, you no longer saw the significant RPE degeneration in the model. I can think of in an analogous way, being interested in the same thing here, is the are the fats that are triggering this hydroxyacylcarnitine accumulation primarily coming from the serum or primarily coming from the load, the photoreceptor outer segment load? And a cross like that would be interesting. Of course, that's easy to say. That's a long multi-year project, but I mean, I'm just immediately curious to know, whether the primary burden to the RPE in this case is from the photoreceptors or from just serum lipid uptake.
I think that's a really critical question. We were approaching it a little bit differently. So we're in the process of doing liver-directed gene addition in the mice to try to lower it in circulation and then see if we still see the retinal phenotype in the mouse. So that's that's at least how my initial approach was, because, if you do liver or muscle-directed gene therapy, you might correct some of the other complications of the disease.
And then the circulating, in that you're saying your hypothesis there would be that would test whether the circulating hydroxyacylcarnitines are causing a unique problem in the RPE,
right?
Or do you need local production?
Local production, right? So separating out, do you have to go to the eye to treat it, the retinal phenotype, or if you lower the circulating levels, do you correct it? So we're going to try it in the mouse I think most of them are coming from the liver. Not 100% sure, but if we correct the liver, can we lower the circulating levels, and then do we see the same retinal degeneration in the eye?
And just to add to that conversation, I mean, maybe this is your motivation for going to the liver first, but with decompensation, you get worse eye results, right? And that decompensation shouldn't theoretically affect the eye via outer segments.
Yes. So the more, the finished group has actually shown that kids who have more metabolic decompensation have a much more rapid loss of vision. And I would argue that each time they decompensate, their hydroxyacylcarnitines are through the roof. So they're in the hospital, they have really high hydroxys. We start, glucose drips and try to get the switch from catabolism to anabolism, and that lowers them, but it takes some time. So I would argue that they're the same, but the finished group, when you've got socialized medicine and really count how many hospitalizations you have, you can really show that the more hospitalizations are related to greater vision loss. So yes, that would be the argument.
I have several questions, and I don't want to stop anyone from, I don't have like crazy amount. Just if anyone else wants to go first, though, I want to give them the opportunity because I've talked plenty.
Yeah. So yeah, very good talk, Melanie. Very cool story. So this is going to be left field, and I'm probably going to expose my massive underknowledge of physiology. So medical doctors, please correct me. But one of the interesting things is that growth hormone starts to decrease very early in life. And its role in adipose tissue is fatty acid release. I was wondering if it does something similar in the RPE, and maybe this is why there's a correlation in the onset of the disease with age.
Puberty.
Have you ever explored that, or has anybody looked at that?
Not that I'm aware of. I'm trying to think if I know of a citation where we've looked at growth hormone. They grow pretty normally, so I don't know that that's been on the radar to look at the growth hormone axis.
Oh, sorry, yeah. So growth hormone just goes down naturally as you age. So it peaks very early in life and then starts going down naturally, which is where you start seeing the disease. So I was wondering if it's protective and the decrease in it may predispose to the onset of symptoms.
Yeah, that's really interesting. No, to my knowledge, nobody's looked at that.
I'll follow up with a related question. With the age of onset coinciding with puberty, do you think that it's hormonal changes in circulating fatty acid metabolism that could be responsible for anything here? I don't really know what those changes would be.
Certainly possible. So it seems to correlate with this growth spurt, right? So their caloric needs go up. And if you're not balancing that with intake, then your hydroxyacylcarnitines would go up. We see a progression in the peripheral neuropathy as well with the onset of puberty. So at the onset of puberty, you'll see a lot of patients who will become wheelchair bound for a period of time. And then it seems to reverse. I still don't fully understand the neuropathy. They have foot drop, need assistance, fall down a lot, be wheelchair bound, and then 6 to 12 months later they'll get up and start walking again. So it seems to be sort of this waxing and waning, whereas the retinopathy seems to be a steady downward trajectory.
Does that point to like some sort of temporary inflammatory response that goes away?
Possibly. We've sort of hypothesized that it's myelination, and their nerves are growing and lengthening, and they're trying to catch up with the myelination, but people haven't done. Go ahead.
You also get you get peripheral nerve regeneration, but you don't get if an RPE is gone,
it's gone.
No regeneration of the I mean, there's some controversy, but most people don't think that you get any regeneration of the RPE.
Right.
So the same insult could have the recovery in one tissue type and a permanent
and not in the other, right?
Okay, I have at least two, maybe. Yeah, two questions. The first, when the RPE are given outer segments, the LCHADD RPE, do you see increased 3-hydroxyacylcarnitines then? I mean, that would again sort of point to this general question of retina-derived versus systemic-derived.
That's a wonderful question. I would assume so, but I'm not sure that we've directly measured that. Like fed the POS and then taken the media and measured hydroxyacylcarnitines. We can certainly do that and add that to our growing list of experiments we need to do. But I don't know that we directly measured that.
Okay. And then just thinking about this, you were saying in the beginning that some patients will take short chain fatty acids.
Medium chain, yeah.
Medium chain, sorry. And so those will never, I mean, there are short chain hydroxyacylcarnitines out there, right? But is the argument that there will be fewer because they don't need carnitine acylation?
Well, so the medium chain fats are oxidized by MCAD. So what I've shown you in the pathway is only the long chain fats. So they bypass all those enzymes. So they would go through MCAD. Then there's a whole set of medium chain enzymes. So they can burn those for fat. And I've done lots of experiments where you give MCT and you can see the increase in oxygen consumption. You can see improved exercise, on a treadmill. So MCT, they can definitely burn. And if you give MCT, you see this big bump in circulating ketones. So they can take MCT in the liver and convert them to ketones where they can't do that with long chain. So it's really just bypassing the block in the long chain
yeah,
pathway.
wonderful.
Yeah.
Okay, that is everything I have, and it looks like some people are leaving anyway. Anyone else have any questions? If not, then thank you so much for the wonderful talk.
Thanks for presenting.
Yeah, thank you. It was fun.
Thank you very much.